The Shroud of Turin. That's the linen cloth believed to bear Jesus' imprint. Now there's new research that may disprove the claim.
For more than 500 years, the Shroud of Turin has remained one of the world's most mysterious and debated artifacts. Some believe it's the real burial cloth of Jesus Christ. Others believe it's nothing more than an elaborate medieval forgery. But in 2015, scientists from the University of Padua discovered something hidden deep inside its ancient linen fibers, microscopic DNA that challenged nearly every theory about the shroud. But that wasn't all. When forensic experts re-examined the red stains on the cloth, they concluded they were real human blood, showing signs of extreme physical trauma. So, are all these independent scientific findings really suggesting that this shroud once wrapped the body of Jesus Christ? Or is this one of the greatest coincidences in scientific history? Let's find out. The image that defied history. But before we talk about the DNA test and the results that stunned scientists, we have to start with the image itself, the same faint image you've probably seen countless times online. And for that, we need to go back to one night nearly 128 years ago. May 28, 1898. Inside the Cathedral of Turin, an Italian lawyer and amateur photographer named Secondo Pia was given a rare opportunity. With permission from King Umberto I, the legal owner of the shroud through the House of Savoy, Pia was allowed to photograph the famous cloth during a public exhibition. At the time, it seemed like nothing more than another photography job. No one imagined it would become the moment that changed this mystery forever. Back then, photography wasn't simple. Pia worked with a massive wooden camera fitted with a Voigtländer lens, and instead of film, he used heavy 50 by 60 centimeter glass plates coated with a light-sensitive chemical. The cathedral was so dark that he had to rely on explosive magnesium flash powder just to light the room. After a few failed attempts, he finally managed to capture 2 successful photographs. But little did he know, the hardest part of the night was still ahead. Back in his dark room, Pia carefully lowered one of the glass plates into a tray of developing chemicals. As the image slowly appeared through the liquid, he reportedly froze. His hands shook so badly that he almost dropped the plate, because what was staring back at him simply wasn't supposed to exist. Normally, a camera creates a negative, where light areas become dark and dark areas become light. But this time, the opposite happened. The faint image printed on the shroud suddenly became a detailed, lifelike portrait of a man. His eyes appeared swollen shut. One cheek looked badly bruised. His nose seemed broken and pushed to one side. Even his beard appeared split down the middle. Details that were barely visible on the linen suddenly stood out with astonishing clarity. And that's where the real mystery began. If the shroud had really been created by a medieval artist, how could someone paint an image that only looks correct when it's photographed as a negative? Nearly 600 years before cameras even existed, there was no way to see the final result, no understanding of photographic negatives, and no method to check whether the light and shadows had been reversed correctly. Yet somehow, they appeared exactly where they should be. Suddenly, the mystery wasn't whether the cloth looked unusual, it was how anyone could have created an image like this at all. For decades, researchers searched for that answer. But as science became more advanced, they realized they had been looking in the wrong place. The biggest clue wasn't printed on the surface of the cloth. It was buried deep between its ancient linen fibers, where centuries of history had been quietly waiting to be discovered. Opening the time capsule. October 5, 2015. More than a century had passed since Secondo Pia's famous photographs. On that day, Professor Gianni Barcaccia and his team at the University of Padua published a groundbreaking DNA study on the Shroud of Turin in the journal Scientific Reports. And unlike any investigation before it, this one wasn't trying to explain the mysterious image. It was trying to answer a different question altogether. What secrets had the cloth itself been carrying for centuries? To find the answer, they didn't cut pieces from the fabric. Instead, they used specially designed sterile micro-vacuum devices fitted with ultra-fine filters. These devices gently collected microscopic dust, pollen, tiny skin flakes, animal hairs, and other particles trapped deep inside the cloth. And there was a reason those traces had survived for so long. The shroud is woven from vertical and horizontal linen threads, known as the warp and weft. Over hundreds of years, the constant rubbing between those threads created tiny pockets where microscopic particles could become sealed away, almost like nature's own evidence bags. Every person who touched the cloth, every place it traveled, had the potential to leave behind another invisible clue. But collecting those particles was only the beginning. The team focused on mitochondrial DNA, a type of DNA found in thousands of copies inside every cell. Unlike ordinary DNA, it survives much longer in ancient material, making it one of the best tools for studying objects that are hundreds or even thousands of years old. It can also help trace where a person's maternal ancestors came from. Over the following weeks, powerful computers decoded millions of nucleotide sequences, the tiny chemical letters that make up DNA. Every fragment was compared with massive genetic databases containing samples from populations around the world. Now, before the results came back, the scientists believed there were only two realistic possibilities. If the shroud was a medieval forgery, the DNA should be mostly European. If it truly originated in the Middle East, that region should dominate the genetic map. The test was expected to settle one of history's longest-running debates. Then the computers finished the analysis. The final genetic map appeared on the screen, and the room went silent. Neither prediction was right. Instead of pointing to one person, or even one region, the shroud seemed to carry the fingerprints of an entire world. The impossible DNA map. The first surprise came from the Middle East. The strongest genetic signatures matched ancient maternal lineages still found across the Levant, especially among the Druze, a close-knit community living mainly in present-day Israel, Lebanon, Syria, and Jordan. Because the Druze have rarely married outside their own community for centuries, their DNA has changed very little over time. Finding those genetic markers strongly suggested that the shroud had a deep connection to the very region where Jesus lived. So far, everything still made sense. Then the map expanded. The computers identified major genetic lineages from Western Europe, including haplogroups U5b, H1, H2, and H3. At first, that wasn't especially surprising either. The shroud had spent hundreds of years in Europe. It had passed through the hands of the House of Savoy, been repaired by the Poor Clare nuns after the fire of 1532, and been viewed by countless pilgrims. Those centuries of contact could easily explain the European DNA. But then, the next result appeared. Scientists found ancient African lineages, including L3, a genetic branch linked mainly to Egypt and Ethiopia. That was unexpected, but still possible. Egypt had traded with the Middle East for thousands of years. Moments later, the map expanded again. This time, it revealed DNA from the Indian subcontinent, including haplogroups M39, M56, and R8. Now the pattern was becoming much harder to explain. But then came the biggest surprise of all. The computers identified genetic markers commonly found in China and Japan, including D4 and G2A. By this point, the map was no longer pointing to one region or even one continent. It seemed to trace a journey spanning much of the ancient world. And to understand why this mattered, you first need to know one simple thing. These genetic lineages, known as haplogroups, act like permanent markers passed down through generations. They don't identify one specific person. Instead, they help show where someone's maternal ancestors came from thousands of years ago. And the map forming on the researchers' computers wasn't pointing to one family or even one country. It was tracing a journey across continents. That left only one possible explanation. The DNA hadn't been placed on the cloth by one person. It had built up naturally over centuries. tiny flakes of skin, loose hairs, dried sweat, and even microscopic droplets left behind by kings, merchants, nuns, priests, and millions of pilgrims had slowly become trapped inside the linen. The shroud wasn't carrying the DNA of a single individual, it had become a biological record of everyone who had crossed its path. The DNA answered one question, but it immediately raised another, because trapped inside the same ancient linen were hundreds of microscopic pollen grains. And once botanists identified where they came from, the mystery became even harder to explain. Plants from another world. More than 40 years before the DNA study, another group of scientists had already begun collecting evidence from the shroud, evidence that had nothing to do with genetics. In 1973, Swiss forensic scientist Max Frey pressed special adhesive tape against the cloth to collect microscopic particles trapped on its surface. He repeated the process again in 1978, preserving tiny pollen grains that were invisible to the naked eye. Years later, those samples were re-examined by Professor Avinoam Danon, a world-renowned botanist from the Hebrew University of Jerusalem. Together, their work identified 58 different plant species trapped within the linen. Only 17 were native to Europe. Most of the others came from the Middle East, the Eastern Mediterranean, and parts of modern-day Turkey. And unlike dust, pollen is remarkably difficult to destroy. Each grain is protected by an incredibly tough outer shell called sporopollenin, allowing it to survive for thousands of years. In other words, these weren't just tiny grains, they were microscopic time capsules. Then came the most surprising discovery. One of the most common pollen types belonged to Gundelia tournefortii, a thorny desert plant that grows mainly in Israel, Jordan, and Syria. Even more interesting, large amounts of its pollen were found around the head area of the image. The plant blooms between March and May, the same season as Passover, leading some researchers to suggest it could have been connected to the crown of thorns described in the Gospel accounts. Scientists also identified pollen from Zygophyllum dumosum, a desert shrub that grows only in Israel, the Judean desert, and parts of the Sinai Peninsula. It simply doesn't grow in the cold, damp climate of medieval France. On its own, none of this proves the shroud belonged to Jesus. But that's not what caught scientists' attention. What mattered was that this completely separate line of evidence pointed to the same places the DNA had already suggested. Two different investigations, using two completely different methods, were beginning to tell the same story. And if the plants pointed to where the cloth had been, the stains on the linen raised an even more disturbing question. What had happened to the man wrapped inside it? What the blood revealed? For years, one of the biggest arguments against the shroud was surprisingly simple. Skeptics believed the reddish stains weren't blood at all. They were just paint applied by a medieval artist. If that were true, the entire story would fall apart. In 2017, scientists finally decided to test that claim using technology far beyond anything available in the past. Professor Giulio Fanti and his team at the University of Padua, working with medical experts from the University Hospital of Trieste, re-examined tiny samples from the stains. They used transmission electron microscopy, which can reveal structures at an almost atomic level, along with Raman spectroscopy, a technique that identifies substances by their unique molecular fingerprints without damaging the sample. The results challenged one of the oldest criticisms of the shroud. The stains were identified as real human blood, belonging to the relatively rare AB blood group. Even more interesting, the same blood type has also been reported on the Sudarium of Oviedo, a cloth preserved in Spain that, according to tradition, covered the face of the same crucified man before burial. But the blood itself wasn't the biggest surprise. Under the microscope, researchers found tiny clusters made up of hemoglobin, ferritin, and creatinine. You don't need to remember those names. What matters is what they mean. Together, they form a biochemical pattern associated with extreme physical trauma. One possible explanation is a condition called rhabdomyolysis. It happens when muscles are damaged so severely that they begin breaking down, flooding the bloodstream with proteins and waste products. Doctors usually see it after crushing injuries, prolonged torture, or other catastrophic trauma. When researchers mapped the blood stains across the body, they also identified injuries consistent with repeated blows from a Roman flagrum, a whip fitted with leather straps, and small lead weights designed to tear deep into the skin. Some forensic studies estimate more than 100 wounds across the back, shoulders, and legs. Then there was one final puzzle. Ancient blood normally turns dark brown or almost black over time. Yet the stains on the shroud have remained noticeably reddish. Researchers believe unusually high levels of bilirubin, a substance released into the blood during severe trauma and intense physical stress, may help explain why. Whether one accepts those conclusions or not, the stains no longer looked like simple paint. And just when the evidence seemed to be pointing in one direction, another test appeared to erase everything with a single date. The age question. For many people, none of the evidence we've looked at mattered anymore after 1988. That was the year 3 of the world's leading laboratories, the University of Oxford, the Swiss Federal Institute of Technology in Zurich, and the University of Arizona, carried out the first official radiocarbon dating test on the shroud. The results seemed clear. All 3 laboratories reached almost the same conclusion. The cloth dated to somewhere between 1260 and 1390 AD, placing it squarely in the Middle Ages, around the same time it first appeared in historical records in France. For much of the scientific community, the case was over. The shroud was declared a medieval creation, rather than a first-century burial cloth. But not everyone was convinced. One of the people who questioned the result was Raymond Rogers, a respected chemist from Los Alamos National Laboratory and a member of the Shroud of Turin research project, also known as STURP. Instead of challenging the dating method itself, Rogers examined something much simpler, the piece of cloth that had been tested. His analysis suggested that the sample had been taken from a repaired corner rather than the original fabric. He found traces of cotton fibers woven into the linen, along with alizarin dye and gum arabic, materials commonly used in medieval textile repairs to blend new threads with old ones. If Rogers was right, the laboratories hadn't dated the original shroud at all. They had dated a later repair. That idea remained controversial for years. Then, in 2022, Dr. Liberato De Caro, from Italy's Institute of Crystallography, approached the problem using a completely different method. Instead of measuring radioactive carbon, he examined the natural aging of the linen itself through wide-angle X-ray scattering, or WAXS. Over time, the cellulose inside linen slowly changes its structure, creating what researchers describe as a kind of molecular clock. After comparing the shroud with ancient textiles of known ages, including Egyptian mummy wrappings and linen recovered from the fortress of Masada in Israel, De Caro reached a very different conclusion. The molecular structure matched fabrics dating to roughly 55 to 74 AD, placing the cloth in the same time period as the life of Jesus. And now, when every question about the shroud seemed to settle down, the same question Secondo Pia unknowingly raised on the night of May 28, 1898, still remains. How was the image itself created? The final mystery. Right now, even after so many investigations and scientific tests, there is still no definitive answer to how that faint image was created. But thanks to advances in science, what we do know about the image is far more than anyone once thought possible. Under the microscope, researchers found that the image isn't soaked into the cloth like paint or ink. It exists only on the outer surface of the linen fibers at a depth of about 200 nanometers, around 500 times thinner than a human hair. Chemical analysis found no paint, no dye, and no pigments. Instead, the fibers appear to have undergone a tiny chemical change caused by oxidation and dehydration, almost like a microscopic scorch that affected only the very surface. Then there was another surprise. In 1976, researchers tested the shroud using NASA's VP-8 image analyzer, a device originally built to turn ordinary images into three-dimensional maps. When normal photographs and paintings were processed, the results became distorted and unnatural. But when the shroud was analyzed, something unexpected happened. The machine produced a recognizable 3D shape of a human body. The reason was even more unusual. The brightness of the image seemed to contain information about the distance between the cloth and the body beneath it, almost like a hidden topographical map recorded on the fabric. Researchers have also noticed several other unusual details. Some believe they can see faint outlines of small bronze coins over the eyes, similar to coins minted during the rule of the Roman governor Pontius Pilate around 29 AD. Others point out that the wounds appear on the wrists instead of the palms, which matches what modern forensic studies say about Roman crucifixion. Even the missing thumbs may have an explanation. A nail driven through the wrist could damage the median nerve, causing the thumbs to naturally curl into the palms. Taken individually, each of these findings has been debated, but together they tell a remarkably consistent story. Different branches of science, working independently, have described the same man, a man who appears to have suffered injuries consistent with Roman crucifixion in 1st century Judea. So, after hearing all of this, what do you think? Is the Shroud of Turin simply an extraordinary ancient artifact that science has not fully explained yet? Or could it really be connected to one of the most debated events in human history? Let us know your thoughts in the comments below. If you enjoyed this video, don't forget to give it a thumbs up. And if you want more incredible mysteries, discoveries, and evidence-based stories like this, be sure to subscribe to the channel. Thanks for watching, and we'll see you in the next video.